Heritability. Heritability is a statistic used in the fields of breeding and genetics that estimates the degree of variation in a phenotypic trait in a population that is due to genetic variation between individuals in that population. 1. The concept of heritability can be expressed in the form of the following question: what is the proportion of the variation in a given trait within a population that is not explained by the environment or random chance? 2. Studies of heritability ask questions such as to what extent do genetic factors influence differences in height between people. This is not the same as asking to what extent do genetic factors influence height in any one person. Other causes of measured variation in a trait are characterized as environmental factors, including observational error. In human studies of heritability, these are often apportioned into factors from shared environment and non-shared environment, based on whether they tend to result in persons brought up in the same household being more or less similar to persons who were not. Heritability is estimated by comparing individual phenotypic variation among related individuals in a population, by examining the association between individual phenotypes. Studies of heritability ask questions such as to what extent do genetic factors influence differences in height between people. This is not the same as asking to what extent do genetic factors influence height in any one person. Other causes of measured variation in a trait are characterized as environmental factors, including observational error. In human studies of heritability, these are often apportioned into factors from shared environment and non-shared environment, based on whether they tend to result in persons brought up in the same household being more or less similar to persons who were not. Heritability is estimated by comparing individual phenotypic variation among related individuals in a population, by examining the association between individual phenotype and genotype data, 3, 4, or even by modeling summary-level data from genome-wide association studies (GWAS). 5. Heritability is an important concept in quantitative genetics, particularly in selective breeding and behavior genetics, for instance, twin studies. It is the source of much confusion due to the fact that its technical definition is different from its commonly understood folk definition. Therefore, its use conveys the incorrect impression that behavioral traits are inherited or specifically passed down through the genes. 6. Behavioral geneticists also conduct heritability analyses based on the assumption that genes and environments contribute in a separate, additive manner to behavioral traits. 7. Overview. Heritability measures the fraction of phenotype variability that can be attributed to genetic variation. This is not the same as saying that this fraction of an individual phenotype is caused by genetics. For example, it is incorrect to say that since the heritability of personality traits is about 0.6, that means that 60% of your personality is inherited from your parents and 40% comes from the environment. In addition, heritability can change without any genetic change occurring, such as when the environment starts contributing to more variation. Heritability. Heritability is a statistic used in the fields of breeding and genetics that estimates the degree of variation in a phenotypic trait in a population that is due to genetic variation between individuals in that population. 1. The concept of heritability can be expressed in the form of the following question: what is the proportion of the variation in a given trait within a population that is not explained by the environment or random chance? 2. Studies of heritability ask questions such as to what extent do genetic factors influence differences in height between people. This is not the same as asking to what extent do genetic factors influence height in any one person. Other causes of measured variation in a trait are characterized as environmental factors, including observational error. In human studies of heritability, these are often apportioned into factors from shared environment and non-shared environment, based on whether they tend to result in persons brought up in the same household being more or less similar to persons who were not. Heritability is estimated by comparing individual phenotypic variation among related individuals in a population, by examining the association between individual phenotype and genotype data, 3, 4, or even by modeling summary-level data from genome-wide association studies (GWAS). 5. Heritability is an important concept in quantitative genetics, particularly in selective breeding and behavior genetics, for instance, twin studies. It is the source of much confusion due to the fact that its technical definition is different from its commonly understood folk definition. Therefore, its use conveys the incorrect impression that behavioral traits are inherited or specifically passed down through the genes. 6. Behavioral geneticists also conduct heritability analyses based on the assumption that genes and environments contribute in a separate, additive manner to behavioral traits. 7. Overview. Heritability measures the fraction of phenotype variability that can be attributed to genetic variation. This is not the same as saying that this fraction of an individual phenotype is caused by genetics. For example, it is incorrect to say that since the heritability of personality traits is about 0.6, that means that 60% of your personality is inherited from your parents and 40% comes from the environment. In addition, heritability can change without any genetic change occurring, such as when the environment starts contributing to more variation. As a case in point, consider that both genes and environment have the potential to influence intelligence. Heritability could increase if genetic variation increases, causing individuals. Heritability measures the fraction of phenotype variability that can be attributed to genetic variation. This is not the same as saying that this fraction of an individual phenotype is caused by genetics. For example, it is incorrect to say that since the heritability of personality traits is about 0.6, that means that 60% of your personality is inherited from your parents and 40% comes from the environment. In addition, heritability can change without any genetic change occurring, such as when the environment starts contributing to more variation. As a case in point, consider that both genes and environment have the potential to influence intelligence. Heritability could increase if genetic variation increases, causing individuals to show more phenotypic variation, like showing different levels of intelligence. On the other hand, heritability might also increase if the environmental variation decreases, causing individuals to show less phenotypic variation, like showing more similar levels of intelligence. Heritability increases when genetics are contributing more variation or because non-genetic factors are contributing less variation. What matters is the relative contribution. Heritability is specific to a particular population in a particular environment. High heritability of a trait, consequently, does not necessarily mean that the trait is not very susceptible to environmental influences. 8. Heritability can also change as a result of changes in the environment, migration, inbreeding, or how heritability itself is measured in the population under study. 9. The heritability of a trait should not be interpreted as a measure of the extent to which said trait is genetically determined in an individual. 10. 11. The extent of dependence of phenotype on environment can also be a function of the genes involved. Matters of heritability are complicated because genes may canalize a phenotype, making its expression almost inevitable in all occurring environments. Individuals with the same genotype can also exhibit different phenotypes through a mechanism called phenotypic plasticity, which makes heritability difficult to measure in some cases. Recent insights in molecular biology have identified changes in transcriptional activity of individual genes associated with environmental changes. However, there are a large number of genes whose transcription is not affected by the environment. 12. Estimates of heritability use statistical analyses to help to identify the causes of differences between individuals. Since heritability is concerned with variance, it is necessarily an account of the differences between individuals in a population. Heritability can be univariate, examining a single trait, or multivariate, examining the genetic and environmental associations between multiple traits at once. This allows a test of the genetic overlap between different phenotypes, for instance hair color and eye color. Environment and genetics may also interact, and heritability analyses can test for and examine these interactions GXE models. A prerequisite for heritability analyses is that there is some population variation to account for. This last point highlights the fact that heritability cannot take into account the effect of factors which are invariant in the population. Factors may be invariant if they are absent and do not exist in the population, such as no one having access to a particular antibiotic, or because they are omnipresent, like if everyone is drinking coffee. In practice, all human behavioral traits vary and almost all traits show some heritability. 13. Definition. Any particular phenotype can be modeled as the sum of genetic and environmental effects. 14. Phenotype P equals genotype G plus environment E. Likewise, the phenotypic variance in the trait, var P, is the sum of effects as follows. Var P equals var G plus var E plus 2 COV G, E. In a planned experiment COV G, E can be controlled and held at 0. In this case, heritability is defined as 15. H2 is the broad sense heritability. This reflects all the genetic contributions to a population's phenotypic variance including additive, dominant, and epistatic multigenic interactions, as well as maternal and paternal effects, where individuals are directly affected by their parents' phenotype, such as with milk production in mammals. A particularly important component of the genetic variance is the additive variance, var A, which is the variance due to the average effects, additive effects, of the alleles. Since each parent passes a single allele per locus to each offspring, parent-offspring resemblance depends upon the average effect of single alleles. Additive variance represents, therefore, the genetic component of variance responsible for parent-offspring resemblance. The additive genetic portion of the phenotypic variance is known as narrow sense heritability and is defined as an uppercase H2 is used to denote broad sense, and lowercase h2 for narrow sense. For traits which are not continuous but dichotomous such as an additional toe or certain diseases, the contribution of the various alleles can be considered to be a sum, which past the threshold, manifests itself as the trait, giving the liability threshold model in which heritability can be estimated and selection modeled. Additive variance is important for selection. If a selective pressure such as improving livestock is exerted, the response of the trait is directly related to narrow sense heritability. The mean of the trait will increase in the next generation as a function of how much the mean of the selected parents differs from the mean of the population from which the selected parents were chosen. The observed response to selection leads to an estimate of the narrow sense heritability called realized heritability. This is the principle underlying artificial selection or breeding. Example. Figure 1. Relationship of phenotypic values to additive and dominance effects using a completely dominant locus. The simplest genetic model involves a single locus with two alleles B and B affecting one quantitative phenotype. The number of B alleles can be 0, 1, or 2. For any genotype, BI, BJ, where BI and BJ are either 0 or 1, the expected phenotype can then be written as the sum of the overall mean, a linear effect, and a dominance deviation. One can think of the dominance term as an interaction between BI and BJ. The additive genetic variance at this locus is the weighted average of the squares of the additive effects. Where there is a similar relationship for the variance of dominance deviations. Where the linear regression of phenotype on genotype is shown in Figure 1. Assumptions. Estimates of the total heritability of human traits assume the absence of epistasis, which has been called the assumption of additivity. Although some researchers have cited such estimates in support of the existence of missing heritability, unaccounted for by known genetic loci, the assumption of additivity may render these estimates invalid. 16. There is also some empirical evidence that the additivity assumption is frequently violated in behavior genetic studies of adolescent intelligence and academic achievement. 17. Estimating heritability. Since only P can be observed or measured directly, heritability must be estimated from the similarities observed in subjects varying in their level of genetic or environmental similarity. The statistical analyses required to estimate the genetic and environmental components of variance depend on the sample characteristics. Briefly, better estimates are obtained using data from individuals with widely varying levels of genetic relationships such as twins, siblings, parents and offspring, rather than from more distantly related, and therefore less similar, subjects. The standard error for heritability estimates is improved with large sample sizes. In non-human populations it is often possible to collect information in a controlled way. For example, among farm animals it is easy to arrange for a bull to produce offspring from a large number of cows into control environments. Such experimental control is generally not possible when gathering human data, relying on naturally occurring relationships and environments. In classical quantitative genetics, there were two schools of thought regarding estimation of heritability. One school of thought was developed by Sewell Wright at the University of Chicago, and further popularized by C. C. Lee, University of Chicago, and J. L. Lush, Iowa State University. It is based on the analysis of correlations and, by extension, regression. Path analysis was developed by Sewell Wright as a way of estimating heritability. The second was originally developed by R. A. Fisher and expanded at the University of Edinburgh, Iowa State University, and North Carolina State University, as well as other schools. It is based on the analysis of variance of breeding studies, using the interclass correlation of relatives. Various methods of estimating components of variance and, hence, heritability from ANOVA are used in these analyses. Today, heritability can be estimated from general pedigrees using linear mixed models and from genomic relatedness estimated from genetic markers. Studies of human heritability often utilize adoption study designs, often with identical twins who have been separated early in life and raised in different environments. Such individuals have identical genotypes and can be used to separate the effects of genotype and environment. A limit of this design is the common prenatal environment and the relatively low numbers of twins reared apart. A second and more common design is the twin study in which the similarity of identical and fraternal twins is used to estimate heritability. These studies can be limited by the fact that identical twins are not completely genetically identical, potentially resulting in an underestimation of heritability. In observational studies, or because of evocative effects where a genome evokes environments by its effect on them, G and E may covary, gene-environment correlation. Depending on the methods used to estimate heritability, correlations between genetic factors and shared or non-shared environments may or may not be confounded with heritability. 18. Regression correlation methods of estimation. The first school of estimation uses regression and correlation to estimate heritability. Comparison of close relatives. In the comparison of relatives, we find that in general, where R can be thought of as the coefficient of relatedness, B is the coefficient of regression and T is the coefficient of correlation. Parent-offspring regression. Figure 2. Francis Galton's 1889 data showing the relationship between offspring height, 928 individuals, as a function of mean parent height, 205 sets of parents. Heritability may be estimated by comparing parent and offspring traits, as in Figure 2. The slope of the line, 0.57, approximates the heritability of the trait when offspring values are regressed against the average trait in the parents. If only one parent's value is used, then heritability is twice the slope. This is the source of the term, regression, since the offspring values always tend to regress to the mean value for the population, i.e., the slope is always less than 1. This regression effect also underlies the de Vries-Volcker method for analyzing twins selected for one member being affected. 19. Sibling comparison. A basic approach to heritability can be taken using full SIB designs, comparing similarity between siblings who share both a biological mother and a father. 20. When there is only additive gene action, this sibling phenotypic correlation is an index of familiarity, the sum of half the additive genetic variance plus full effect of the common environment. It thus places an upper limit on additive heritability of twice the full SIB phenotypic correlation. Half SIB designs compare phenotypic traits of siblings that share one parent with other sibling groups. Twin studies. Figure 3. Twin concordances for 7 psychological traits, sample size shown in sidebars, with DZ being fraternal and MZ being identical twins. Heritability for traits in humans is most frequently estimated by comparing resemblances between twins. The advantage of twin studies is that the total variance can be split up into genetic, shared or common environmental, and unique environmental components, enabling an accurate estimation of heritability. 21. Fraternal or dizygotic DZ twins on average share half their genes, assuming there is no associative mating for the trait, and so identical or monozygotic MZ twins on average are twice as genetically similar as DZ twins. A crude estimate of heritability, then, is approximately twice the difference in correlation between MZ and DZ twins, i.e. Falconer's formula H2 equals 2 R MZ R DZ. The effect of shared environment, C2, contributes to similarity between siblings due to the commonality of the environment they are raised in. Shared environment is approximated by the DZ correlation minus half heritability, which is the degree to which DZ twins share the same genes, C2 equals DZ 1/2 H2. Unique environmental variance, E2, reflects the degree to which identical twins raised together are dissimilar, E2 equals 1 R MZ. Analysis of variance methods of estimation. The second set of methods of estimation of heritability involves ANOVA and estimation of variance components. Basic model. We use the basic discussion of Kempthorne. 14. Considering only the most basic of genetic models. We can look at the quantitative contribution of a single locus with genotype GI as. Where is the effect of genotype GI and is the environmental effect? Consider an experiment with a group of sires and their progeny from random dams. Since the progeny get half of their genes from the father and half from their random mother, the progeny equation is interclass correlations. Consider the experiment above. We have two groups of progeny we can compare. The first is comparing the various progeny for an individual sire called within sire group. The variance will include terms for genetic variance, since they did not all get the same genotype, and environmental variance. This is thought of as an error term. The second group of progeny are comparisons of means of half-sibs with each other called among sire group. In addition to the error term as in the within sire groups, we have an addition term due to the differences among different means of half-sibs. The interclass correlation is. Since environmental effects are independent of each other, the ANOVA. In an experiment with sires and progeny per sire, we can calculate the following ANOVA, using as the genetic variance and as the environmental variance. Table 1, ANOVA for sire experiment. Source D, F, mean square expected mean square. Between sire groups. Within sire groups. The term is the interclass correlation between half-sibs. We can easily calculate. The expected mean square is calculated from the relationship of the individual's progeny within a sire or all half-sibs, for example, and an understanding of interclass correlations. The use of ANOVA to calculate heritability often fails to account for the presence of gene-environment interactions, because ANOVA has a much lower statistical power for testing for interaction effects than for direct effects. 22. Model with additive and dominance terms. For a model with additive and dominance terms, but not others, the equation for a single locus is where is the additive effect of the ITH allele, is the additive effect of the JTH allele, is the dominance deviation for the IJTH genotype, and is the environment. Experiments can be run with a similar setup to the one given in Table 1. Using different relationship groups, we can evaluate different interclass correlations. Using as the additive genetic variance and as the dominance deviation variance, interclass correlations become linear functions of these parameters. In general, interclass correlation where and are found as P alleles drawn at random from the relationship pair are identical by descent, and P genotypes drawn at random from the relationship pair are identical by descent. Some common relationships and their coefficients are given in Table 2. Table 2, coefficients for calculating variance components. Relationship. Identical twins parent offspring. Half-siblings. Full siblings. First cousins. Double first cousins. Linear mixed models. A wide variety of approaches using linear mixed models have been reported in literature. Via these methods, phenotypic variance is partitioned into genetic, environmental and experimental design variances to estimate heritability. Environmental variance can be explicitly modeled by studying individuals across a broad range of environments, although inference of genetic variance from phenotypic and environmental variance may lead to underestimation of heritability due to the challenge of capturing the full range of environmental influence affecting a trait. Other methods for calculating heritability use data from genome-wide association studies to estimate the influence on a trait by genetic factors, which is reflected by the rate and influence of putatively associated genetic loci, usually single nucleotide polymorphisms, on the trait. This can lead to underestimation of heritability, however. This discrepancy is referred to as missing heritability, and reflects the challenge of accurately modeling both genetic and environmental variance in heritability models. 23. When a large, complex pedigree or another aforementioned type of data is available, heritability and other quantitative genetic parameters can be estimated by restricted maximum likelihood (REML) or Bayesian methods. The raw data will usually have 3 or more data points for each individual, a code for the sire, a code for the dam, and one or several trait values. Different trait values may be for different traits or for different time points of measurement. The currently popular methodology relies on high degrees of certainty over the identities of the sire and dam. It is not common to treat the sire identity probabilistically. This is not usually a problem, since the methodology is rarely applied to wild populations, although it has been used for several wild ungulate and bird populations, and sires are invariably known with a very high degree of certainty in breeding programs. There are also algorithms that account for uncertain paternity. Pedigree models are helpful for entangling confounds such as reverse causality, maternal effects such as the prenatal environment, and confounding of genetic dominance, shared environment, and maternal gene effects. 24.9. Genomic heritability. When genome-wide genotype data and phenotypes from large population samples are available, one can estimate the relationships between individuals based on their genotypes and use a linear mixed model to estimate the variance explained by the genetic markers. This gives a genomic heritability estimate based on the variance captured by common genetic variants. 4. There are multiple methods that make different adjustments for allele frequency and linkage disequilibrium. Particularly, the method called high-definition likelihood (HDL) can estimate genomic heritability using only GWAS summary statistics. 5. Making it easier to incorporate large sample size available in various GWAS meta-analysis. Response to selection. Figure 4. Strength of selection S and response to selection R in an artificial selection experiment, H2 equals RS. In selective breeding of plants and animals, the expected response to selection of a trait with known narrow sense heritability can be estimated using the breeder's equation, 25. In this equation, the response to selection, R, is defined as the realized average difference between the parent generation and the next generation, and the selection differential, S, is defined as the average difference between the parent generation and the selected parents. 14, 1957, 26. For example, imagine that a plant breeder is involved in a selective breeding project with the aim of increasing the number of kernels per ear of corn. For the sake of argument, let us assume that the average ear of corn in the parent generation has 100 kernels. Let us also assume that the selected parents produce corn with an average of 120 kernels per ear. If H2 equals 0.5, then the next generation will produce corn with an average of 0.5, 120 to 100 equals 10 additional kernels per ear. Therefore, the total number of kernels per ear of corn will equal, on average, 110. Observing the response to selection in an artificial selection experiment will allow calculation of realized heritability as in Figure 4. Heritability in the above equation is equal to the ratio only if the genotype and the environmental noise follow Gaussian distributions. Controversies. Heritability estimates. Prominent critics, such as Stephen Rose, 27, J. Joseph, 28, and Richard Bentall, focus largely on heritability estimates in behavioral sciences and social sciences. Bentall has claimed that such heritability scores are typically calculated counterintuitively to derive numerically high scores, that heritability is misinterpreted as genetic determination, and that this alleged bias distracts from other factors that researchers have found more causally important, such as childhood abuse causing later psychosis. 29, 30. Heritability estimates are also inherently limited because they do not convey any information regarding whether genes or environment play a larger role in the development of the trait under study. For this reason, David Moore and David Schenck describe the term, heritability, in the context of behavior genetics as, one of the most misleading in the history of science, and argue that it has no value except in very rare cases. 31. When studying complex human traits, it is impossible to use heritability analysis to determine the relative contributions of genes and environment, as such traits result from multiple causes interacting. 32. In particular, Feldman and Lewontin emphasize that heritability is itself a function of environmental variation. 33. However, some researchers argue that it is possible to disentangle the two. 34. The controversy over heritability estimates is largely via their basis in twin studies. The scarce success of molecular genetic studies to corroborate such population genetic studies conclusions is the missing heritability problem. 35. Eric Durkheim has argued that newer molecular methods have vindicated the conventional interpretation of twin studies. 35. Although it remains mostly unclear how to explain the relations between genes and behaviors. 36. According to Durkheim, both genes and environment are heritable, genetic contribution varies by environment, and a focus on heritability distracts from other important factors. 37. Overall, however, heritability is a concept widely applicable. Heredity. Heredity, also called inheritance or biological inheritance, is the passing on of traits from parents to their offspring, either through asexual reproduction or sexual reproduction. The offspring cells or organisms acquire the genetic information of their parents. Through heredity, variations between individuals can accumulate and cause species to evolve by natural selection. The study of heredity in biology is genetics. Overview. Heredity of phenotypic traits, a father and son with prominent ears and crowns. DNA structure, bases are in the center, surrounded by phosphate sugar chains in a double helix. In humans, eye color is an example of an inherited characteristic. An individual might inherit the brown eye trait from one of the parents. 1. Inherited traits are controlled by genes and the complete set of genes within an organism's genome is called its genotype. 2. The complete set of observable traits of the structure and behavior of an organism is called its phenotype. Traits arise from the interaction of the organism's genotype with the environment. 3. As a result, many aspects of an organism's phenotype are not inherited. For example, suntan skin derives from the interaction between a person's genotype and sunlight. 4. Thus, suntans are not passed on to people's children. However, some people tan more easily than others, due to differences in their genotype. 5. A striking example is people with the inherited trait of albinism, who do not tan at all and are very sensitive to sunburn. 6. Heritable traits are known to be passed from one generation to the next via DNA, a molecule that encodes genetic information. 2. DNA is a long polymer that incorporates 4 types of bases, which are interchangeable. The nucleic acid sequence, the sequence of bases along a particular DNA molecule, specifies the genetic information. This is comparable to a sequence of letters spelling out a passage of text. 7. Before a cell divides through mitosis, the DNA is copied, so that each of the resulting 2 cells will inherit the DNA sequence. A portion of a DNA molecule that specifies a single functional unit is called a gene. Different genes have different sequences of bases. Within cells, the long strands of DNA form condensed structures called chromosomes. Organisms inherit genetic material from their parents in the form of homologous chromosomes, containing a unique combination of DNA sequences that code for genes. The specific location of a DNA sequence within a chromosome is known as a locus. If the DNA sequence at a particular locus varies between individuals, the different forms of this sequence are called alleles. DNA sequences can change through mutations, producing new alleles. If a mutation occurs within a gene, the new allele may affect the trait that the gene controls, altering the phenotype of the organism. 8. However, while this simple correspondence between an allele and a trait works in some cases, most traits are more complex and are controlled by multiple interacting genes within and among organisms. 9-10 Developmental biologists suggest that complex interactions in genetic networks and communication among cells can lead to heritable variations that may underlie some of the mechanics in developmental plasticity and canalization. 11. Recent findings have confirmed important examples of heritable changes that cannot be explained by direct agency of the DNA molecule. These phenomena are classed as epigenetic inheritance systems that are causally or independently evolving over genes. Research into modes and mechanisms of epigenetic inheritance is still in its scientific infancy, but this area of research has attracted much recent activity as it broadens the scope of heritability and evolutionary biology in general. 12 DNA methylation marking chromatin, self-sustaining metabolic loops, gene silencing by RNA interference, and the three-dimensional conformation of proteins, such as prions, are areas where epigenetic inheritance systems have been discovered at the organismic level. 13-14 Heritability may also occur at even larger scales. For example, ecological inheritance through the process of niche construction is defined by the regular and repeated activities of organisms in their environment. This generates a legacy of effect that modifies and feeds back into the selection regime of subsequent generations. Descendants inherit genes plus environmental characteristics generated by the ecological actions of ancestors. 15 Other examples of heritability in evolution that are not under the direct control of genes include the inheritance of cultural traits, group heritability, and symbiogenesis. 16, 17, 18 These examples of heritability that operate above the gene are covered broadly under the title of multilevel or hierarchical selection, which has been a subject of intense debate in the history of evolutionary science. 17, 19. Relation to theory of evolution. When Charles Darwin proposed his theory of evolution in 1859, one of its major problems was the lack of an underlying mechanism for heredity. 20. Darwin believed in a mix of blending inheritance and the inheritance of acquired traits, pangenesis. Blending inheritance would lead to uniformity across populations in only a few generations and then would remove variation from a population on which natural selection could act. 21. This led to Darwin adopting some Lamarckian ideas in later editions of On the Origin of Species in his later biological works. 22. Darwin's primary approach to heredity was to outline how it appeared to work, noticing that traits that were not expressed explicitly in the parent at the time of reproduction could be inherited, that certain traits could be sex-linked, etc., rather than suggesting mechanisms. Darwin's initial model of heredity was adopted by, and then heavily modified by, his cousin Francis Galton, who laid the framework for the biometric school of heredity. 23. Galton found no evidence to support the aspects of Darwin's pangenesis model, which relied on acquired traits. 24. The inheritance of acquired traits was shown to have little basis in the 1880s when August Weismann cut the tails off many generations of mice and found that their offspring continued to develop tails. 25. History. Aristotle's model of inheritance. The heat-cold part is largely symmetrical, though influenced on the father's side by other factors, but the form part is not. Scientists in antiquity had a variety of ideas about heredity. Theophrastus proposed that male flowers cause female flowers to ripen. 26. Hippocrates speculated that seeds were produced by various body parts and transmitted to offspring at the time of conception. 27. And Aristotle thought that male and female fluids mixed at conception. 28. Aeschylus, in 458 BC, proposed the male as the parent, with the female as a nurse for the young life sown within her. 29. Ancient understandings of heredity transitioned to two debated doctrines in the 18th century. The doctrine of epigenesis and the doctrine of preformation were two distinct views of the understanding of heredity. The doctrine of epigenesis, originated by Aristotle, claimed that an embryo continually develops. The modifications of the parent's traits are passed off to an embryo during its lifetime. The foundation of this doctrine was based on the theory of inheritance of acquired traits. In direct opposition, the doctrine of preformation claimed that like generates like, where the germ would evolve to yield offspring similar to the parents. The preformationist view believed procreation was an act of revealing what had been created long before. However, this was disputed by the creation of the cell theory in the 19th century, where the fundamental unit of life is the cell, and not some preformed parts of an organism. Various hereditary mechanisms, including blending inheritance, were also envisaged without being properly tested or quantified, and were later disputed. Nevertheless, people were able to develop domestic breeds of animals as well as crops through artificial selection. The inheritance of acquired traits also formed a part of early Lamarckian ideas on evolution. During the 18th century, Dutch microscopist Antony van Leeuwenhoek (1632–1723) discovered animalcules in the sperm of humans and other animals. 30. Some scientists speculated they saw a little man, homunculus, inside each sperm. These scientists formed a school of thought known as the spermists. They contended the only contributions of the female to the next generation were the womb in which the homunculus grew, and prenatal influences of the womb. 31. An opposing school of thought, the ovists, believed that the future human was in the egg, and that sperm merely stimulated the growth of the egg. Ovists thought women carried eggs containing boy and girl children, and that the gender of the offspring was determined well before conception. 32. An early research initiative emerged in 1878 when Alpheus Hyatt led an investigation to study the laws of heredity through compiling data on family phenotypes, nose size, ear shape, etc., and expression of pathological conditions and abnormal characteristics, particularly with respect to the age of appearance. One of the project's aims was to tabulate data to better understand why certain traits are consistently expressed while others are highly irregular. 33. Gregor Mendel, father of genetics. Table showing how the genes exchange according to segregation or independent assortment during meiosis and how this translates into Mendel's laws. The idea of particulate inheritance of genes can be attributed to the Moravian, 34, monk Gregor Mendel, who published his work on pea plants in 1865. However, his work was not widely known and was rediscovered in 1901. It was initially assumed that Mendelian inheritance only accounted for large qualitative differences, such as those seen by Mendel in his pea plants, and the idea of additive effect of quantitative genes was not realized until R. A. Fisher's 1918 paper, The Correlation Between Relatives on the Supposition of Mendelian Inheritance, Mendel's Overall Contribution, gave scientists a useful overview that traits were inheritable. His pea plant demonstration became the foundation of the study of Mendelian traits. These traits can be traced on a single locus. 35. Modern development of genetics and heredity. In the 1930s, work by Fisher and others resulted in a combination of Mendelian and biometric schools into the modern evolutionary synthesis. The modern synthesis bridged the gap between experimental geneticists and naturalists, and between both and paleontologists, stating that 36-37. 1. All evolutionary phenomena can be explained in a way consistent with known genetic mechanisms and the observational evidence of naturalists. 2. Evolution is gradual, small genetic changes, recombination ordered by natural selection. Discontinuities amongst species or other taxa are explained as originating gradually through geographical separation and extinction, not saltation. 3. Selection is overwhelmingly the main mechanism of change, even slight advantages are important when continued. The object of selection is the phenotype in its surrounding environment. The role of genetic drift is equivocal, though strongly supported initially by Dobzhansky, it was downgraded later as results from ecological genetics were obtained. 4. The primacy of population thinking. The genetic diversity carried in natural populations is a key factor in evolution. The strength of natural selection in the wild was greater than expected. The effect of ecological factors such as niche occupation and the significance of barriers to gene flow are all important. The idea that speciation occurs after populations are reproductively isolated has been much debated. 38. In plants, polyploidy must be included in any view of speciation. Formulations such as evolution consists primarily of changes in the frequencies of alleles between one generation and another, were proposed rather later. The traditional view is that developmental biology, Evo Devo, played little part in the synthesis, but an account of Gavin de Beer's work by Stephen Jay Gould suggests he may be an exception. 39. Almost all aspects of the synthesis have been challenged at times, with varying degrees of success. There is no doubt, however, that the synthesis was a great landmark in evolutionary biology. 40. It cleared up many confusions, and was directly responsible for stimulating a great deal of research in the post-World War II era. Trofim Lysenko however caused a backlash of what is now called Lysenkoism in the Soviet Union when he emphasized Lamarckian ideas on the inheritance of acquired traits. This movement affected agricultural research and led to food shortages in the 1960s and seriously affected the USSR. 41. There is growing evidence that there is transgenerational inheritance of epigenetic changes in humans, 42, and other animals. 43. Common genetic disorders: Fragile X syndrome, sickle cell disease, phenylketonuria, PKU, hemophilia, 35. Types. An example pedigree chart of an autosomal dominant disorder. An example pedigree chart of an autosomal recessive disorder. An example pedigree chart of a sex-linked disorder. The gene is on the X chromosome. The description of a mode of biological inheritance consists of three main categories. 1. Number of involved loci. Monogenetic, also called simple, one locus; oligogenic, few loci. Polygenetic, many loci. 2. Involved chromosomes. Autosomal, loci are not situated on a sex chromosome. Gonosomal, loci are situated on a sex chromosome. X chromosomal, loci are situated on the X chromosome. The more common case, Y chromosomal, loci are situated on the Y chromosome. Mitochondrial, loci are situated on the mitochondrial DNA. 3. Correlation: genotype, phenotype. Dominant. Intermediate, also called codominant. Recessive. Overdominant. Underdominant. These three categories are part of every exact description of a mode of inheritance in the above order. In addition, more specifications may be added as follows. 4. Coincidental and environmental interactions. Penetrance. Complete. Incomplete percentual number. Expressivity. Invariable. Variable. Heritability in polygenetic and sometimes also in oligogenetic modes of inheritance, maternal or paternal imprinting phenomena, also see epigenetics. 5. Sex-linked interactions. Sex-linked inheritance, gonosomal loci. Sex-limited phenotype expression, e.g., cryptorchism. Inheritance through the maternal line, in case of mitochondrial DNA loci, inheritance through the paternal line, in case of Y chromosomal loci. 6. Locus-locus interactions. Epistasis with other loci, e.g., overdominance. Gene coupling with other loci, also see crossing over. Homozygous lethal factors. Semi-lethal factors. Determination and description of a mode of inheritance is also achieved primarily through statistical analysis of pedigree data. In case the involved loci are known, methods of molecular genetics can also be employed. Dominant and recessive alleles. An allele is said to be dominant if it is always expressed in the appearance of an organism phenotype, provided that at least one copy of it is present. For example, in peas the allele for green pods, G, is dominant to that for yellow pods, G. Thus pea plants with the pair of alleles either GG homozygote or GG heterozygote will have green pods. The allele for yellow pods is recessive. The effects of this allele are only seen when it is present in both chromosomes, GG homozygote. This derives from zygosity, the degree to which both copies of a chromosome or gene have the same genetic sequence, in other words, the degree of similarity of the alleles in an organism. Hereditary defects in enzymes are generally inherited in an autosomal fashion because there are more non-X chromosomes than X chromosomes, and a recessive fashion because the enzymes from the unaffected genes are generally sufficient to prevent symptoms in carriers. On the other hand, hereditary defects in structural proteins, such as osteogenesis imperfecta, Marfan's syndrome and many Ehlers-Danlos syndromes, are generally autosomal dominant, because it is enough that some components are defective to make the whole structure dysfunctional. This is a dominant negative process, wherein a mutated gene product adversely affects the non-mutated gene product within the same cell. Hard inheritance Lamarckism. Heritability. Particulate inheritance. Natural selection. Natural selection is the differential survival and reproduction of individuals due to differences in phenotype. It is a key mechanism of evolution, the change in the heritable traits characteristic of a population over generations. Charles Darwin popularized the term, natural selection, contrasting it with artificial selection, which is intentional, whereas natural selection is not. A diagram demonstrating mutation and selection. Modern biology began in the 19th century with Charles Darwin's work on evolution by natural selection. Variation of traits, both genotypic and phenotypic, exists within all populations of organisms. However, some traits are more likely to facilitate survival and reproductive success. Thus, these traits are passed on to the next generation. These traits can also become more common within a population if the environment that favors these traits remains fixed. If new traits become more favored due to changes in a specific niche, microevolution occurs. If new traits become more favored due to changes in the broader environment, macroevolution occurs. Sometimes, new species can. If new traits become more favored due to changes in a specific niche, microevolution occurs. If new traits become more favored due to changes in the broader environment, macroevolution occurs. Sometimes, new species can arise especially if these new traits are radically different from the traits possessed by their predecessors. The likelihood of these traits being selected and passed down are determined by many factors. Some are likely to be passed down because they adapt well to their environments. Others are passed down because these traits are actively preferred by mating partners, which is known as sexual selection. Female bodies also prefer traits that confer the lowest cost to their reproductive health, which is known as fecundity selection. Natural selection is a cornerstone of modern biology. The concept, published by Darwin and Alfred Russel Wallace in a joint presentation of papers in 1858, was elaborated in Darwin's influential 1859 book On the Origin of Species by Means of Natural Selection, or the Preservation of Favored Races in the Struggle for Life. He described natural selection as analogous to artificial selection, a process by which animals and plants with traits considered desirable by human breeders are systematically favored for reproduction. The concept of natural selection originally developed in the absence of a valid theory of heredity. At the time of Darwin's writing, science had yet to develop modern theories of genetics. The union of traditional Darwinian evolution with subsequent discoveries in classical genetics formed the modern synthesis of the mid-20th century. The addition of molecular genetics has led to evolutionary developmental biology, which explains evolution at the molecular level. While genotypes can slowly change by random genetic drift, natural selection remains the primary explanation for adaptive evolution. Historical development. Pre-Darwinian theories. Aristotle considered whether different forms could have appeared, only the useful ones surviving. Several philosophers of the classical era, including Empedocles I and his intellectual successor, the Roman poet Lucretius II, expressed the idea that nature produces a huge variety of creatures, randomly, and that only those creatures that manage to provide for themselves and reproduce successfully persist. Empedocles' idea that organisms arose entirely by the incidental workings of causes such as heat and cold was criticized by Aristotle in Book 2 of Physics. 3. He posited natural teleology in its place, and believed that form was achieved for a purpose, citing the regularity of heredity in species as proof. 4. 5. Nevertheless, he accepted in his biology that new types of animals, monstrosities terras, can occur in very rare instances. Generation of Animals, Book 4. 6. As quoted in Darwin's 1872 edition of The Origin of Species, Aristotle considered whether different forms, e.g., of teeth might have appeared accidentally, but only the useful forms survived. So what hinders the different parts of the body from having this merely accidental relation in nature? As the teeth, for example, grow by necessity, the front ones sharp, adapted for dividing, and the grinders flat, and serviceable for masticating the food, since they were not made for the sake of this, but it was the result of accident. And in like manner as to the other parts in which there appears to exist an adaptation to an end. Wheresoever, therefore, all things together, that is all the parts of one whole, happened like as if they were made for the sake of something, these were preserved, having been appropriately constituted by an internal spontaneity, and whatsoever things were not thus constituted, perished, and still perish. Aristotle, Physics, Book 2, Chapter 8, 7. But Aristotle rejected this possibility in the next paragraph, making clear that he is talking about the development of animals as embryos with the phrase, either invariably or normally come about, not the origin of species. Yet it is impossible that this should be the true view. For teeth and all other natural things either invariably or normally come about in a given way, but of not one of the results of chance or spontaneity is this true. We do not ascribe to chance or mere coincidence the frequency of rain in winter, but frequent rain in summer we do, nor heat in the dog days, but only if we have it in winter. If then, it is agreed that things are either the result of coincidence or for an end, and these cannot be the result of coincidence or spontaneity, it follows that they must be for an end, and that such things are all due to nature. Even the champions of the theory which is before us would agree. Therefore action for an end is present in things which come to be and are by nature. Aristotle, Physics, Book 2, Chapter 8. The struggle for existence was later described by the Islamic writer Al-Jahiz in the 9th century, particularly in the context of top-down population regulation, but not in reference to individual variation or natural selection. 9-10. At the turn of the 16th century, Leonardo da Vinci collected a set of fossils of ammonites as well as other biological material. He extensively reasoned in his writings that the shapes of animals are not given once and forever by the upper power, but instead are generated in different forms naturally and then selected for reproduction by their compatibility with the environment. 11. The more recent classical arguments were reintroduced in the 18th century by Pierre-Louis Maupertuis, 12, and others, including Darwin's grandfather, Erasmus Darwin. Until the early 19th century, the prevailing view in Western societies was that differences between individuals of a species were uninteresting departures from their Platonic ideals, or typus, of created kinds. However, the theory of uniformitarianism in geology promoted the idea that simple, weak forces could act continuously over long periods of time to produce radical changes in the Earth's landscape. The success of this theory raised awareness of the vast scale of geological time and made plausible the idea that tiny, virtually imperceptible changes in successive generations could produce consequences on the scale of differences between species. 13. The early 19th century zoologist Jean-Baptiste Lamarck suggested the inheritance of acquired characteristics as a mechanism for evolutionary change. Adaptive traits acquired by an organism during its lifetime could be inherited by that organism's progeny, eventually causing transmutation of species. 14. This theory, Lamarckism, was an influence on the Soviet biologist Trofim Lysenko's ill-fated antagonism to mainstream genetic theory as late as the mid-20th century. 15. Between 1835 and 1837, the zoologist Edward Blyth worked on the area of variation, artificial selection, and how a similar process occurs in nature. Darwin acknowledged Blyth's ideas in the first chapter on variation of On the Origin of Species. 16. Darwin's theory. In 1859, Charles Darwin set out his theory of evolution by natural selection as an explanation for adaptation and speciation. He defined natural selection as a principle by which each slight variation of a trait, if useful, is preserved. 17. The concept was simple but powerful. Individuals best adapted to their environments are more likely to survive and reproduce. As long as there is some variation between them and that variation is heritable, there will be an inevitable selection of individuals with the most advantageous variations. If the variations are heritable, then differential reproductive success leads to the evolution of particular populations of a species, and populations that evolve to be sufficiently different eventually become different species. 18-19. Part of Thomas Malthus's Table of Population Growth in England 1780-1810, from his Essay on the Principle of Population, 6th edition, 1826. Darwin's ideas were inspired by the observations that he had made on the second voyage of HMS Beagle 1831-1836, and by the work of a political economist, Thomas Robert Malthus, who, in an essay on the principle of population 1798, noted that population, if unchecked, increases exponentially, whereas the food supply grows only arithmetically. Thus, inevitable limitations of resources would have demographic implications, leading to a struggle for existence. 20. When Darwin read Malthus in 1838, he was already primed by his work as a naturalist to appreciate the struggle for existence in nature. It struck him that as population outgrew resources, favorable variations would tend to be preserved, and unfavorable ones to be destroyed. The result of this would be the formation of new species. 21. Darwin wrote, if during the long course of ages and under varying conditions of life, organic beings vary at all in the several parts of their organization, and I think this cannot be disputed, if there be, owing to the high geometrical powers of increase of each species, at some age, season, or year, a severe struggle for life, and this certainly cannot be disputed, then, considering the infinite complexity of the relations of all organic beings to each other and to their conditions of existence, causing an infinite diversity in structure, constitution, and habits, to be advantageous to them, I think it would be a most extraordinary fact if no variation ever had occurred useful to each being's own welfare, in the same way as so many variations have occurred useful to man. But if variations useful to any organic being do occur, assuredly individuals thus characterized will have the best chance of being preserved in the struggle for life, and from the strong principle of inheritance they will tend to produce offspring similarly characterized. This principle of preservation, I have called, for the sake of brevity, natural selection. Darwin summarizing natural selection in the 4th chapter of On the Origin of Species, 22. 21. Darwin. If during the long course of ages and under varying conditions of life, organic beings vary at all in the several parts of their organization, and I think this cannot be disputed, if there be, owing to the high geometrical powers of increase of each species, at some age, season, or year, a severe struggle for life, and this certainly cannot be disputed, then, considering the infinite complexity of the relations of all organic beings to each other and to their conditions of existence, causing an infinite diversity in structure, constitution, and habits, to be advantageous to them, I think it would be a most extraordinary fact if no variation ever had occurred useful to each being's own welfare, in the same way as so many variations have occurred useful to man. But if variations useful to any organic being do occur, assuredly individuals thus characterized will have the best chance of being preserved in the struggle for life, and from the strong principle of inheritance they will tend to produce offspring similarly characterized. This principle of preservation, I have called, for the sake of brevity, natural selection. Darwin summarizing natural selection in the 4th chapter of On the Origin of Species, 22. Once he had his theory, Darwin was meticulous about gathering and refining evidence before making his idea public. He was in the process of writing. The result of this would be the formation of new species. 21. Darwin wrote. The result of this would be the formation of new species. 21. Darwin wrote. If during the long course of ages and under varying conditions of life, organic beings vary at all in the several parts of their organization, and I think this cannot be disputed, if there be, owing to the high geometrical powers of increase of each species, at some age, season, or year, a severe struggle for life, and this certainly cannot be disputed, then, considering the infinite complexity of the relations of all organic beings to each other and to their conditions of existence, causing an infinite diversity in structure, constitution, and habits, to be advantageous to them, I think it would be a most extraordinary fact if no variation ever had occurred useful to each being's own welfare, in the same way as so many variations have occurred useful to man. But if variations useful to any organic being do occur, assuredly individuals thus characterized will have the best chance of being preserved in the struggle for life, and from the strong principle of inheritance they will tend to produce offspring similarly characterized. This principle of preservation, I have called, for the sake of brevity, natural selection. Darwin summarizing natural selection in the 4th chapter of On the Origin of Species, 22. Once he had his theory, Darwin was meticulous about gathering and refining evidence before making his idea public. He was in the process of writing his big book to present his research when the naturalist Alfred Russel Wallace independently conceived of the principle and described it in an essay he sent to Darwin to forward to Charles Lyell. Lyell and Joseph Dalton Hooker decided to present his essay together with unpublished writings that Darwin had sent to fellow naturalists, and on the tendency of species to form varieties, and on the perpetuation of varieties and species by natural means of selection was read to the Linnean Society of London announcing co-discovery of the principle in July 1858. 23. Darwin published a detailed account of his evidence and conclusions in On the Origin of Species in 1859. In the 3rd edition of 1861 Darwin acknowledged that others, like William Charles Wells in 1813, and Patrick Matthew in 1831, had proposed similar ideas, but had neither developed them nor presented them in notable scientific publications. 24. Charles Darwin noted that pigeon fanciers had created many kinds of pigeon, such as tumblers 1, 12 fantails 13, and pouters 14, by selective breeding. Darwin thought of natural selection by analogy to how farmers select crops or livestock for breeding, which he called artificial selection. In his early manuscripts he referred to it, nature, which would do the selection. At the time, other mechanisms of evolution such as evolution by genetic drift were not yet explicitly formulated, and Darwin believed that selection was likely only part of the story. I am convinced that natural selection has been the main but not exclusive means of modification. 25. In a letter to Charles Lyell in September 1860, Darwin regretted the use of the term natural selection, preferring the term natural preservation. 26. For Darwin and his contemporaries, natural selection was in essence synonymous with evolution by natural selection. After the publication of On the Origin of Species, 27, educated people generally accepted that evolution had occurred in some form. However, natural selection remained controversial as a mechanism, partly because it was perceived to be too weak to explain the range of observed characteristics of living organisms, and partly because even supporters of evolution balked at its unguided and non-progressive nature. 28, a response that has been characterized as the single most significant impediment to the idea's acceptance. 29. However, some thinkers enthusiastically embraced natural selection. After reading Darwin, Herbert Spencer introduced the phrase survival of the fittest, which became a popular summary of the theory. 31. The 5th edition of On the Origin of Species published in 1869 included Spencer's phrase as an alternative to natural selection, with credit given, but the expression often used by Mr. Herbert Spencer of the survival of the fittest is more accurate, and is sometimes equally convenient. 32. Although the phrase is still often used by non-biologists, modern biologists avoid it because it is tautological if fittest is read to mean functionally superior, and is applied to individuals rather than considered as an average quantity over populations. 33. The modern synthesis. Natural selection relies crucially on the idea of heredity, but developed before the basic concepts of genetics. Although the Moravian monk Gregor Mendel, the father of modern genetics, was a contemporary of Darwin's, his work lay in obscurity, only being rediscovered in 1900. 34. With the early 20th century integration of evolution with Mendel's laws of inheritance, the so-called modern synthesis, scientists generally came to accept natural selection. 35. 36. The synthesis grew from advances in different fields. Ronald Fisher developed the required mathematical language and wrote the Genetical Theory of Natural Selection, 1930. 37. J.B.S. Haldane introduced the concept of the cost of natural selection. 38-39. Sewall Wright elucidated the nature of selection and adaptation. 40. In his book Genetics and the Origin of Species, 1937, Theodosius Dobzhansky established the idea that mutation, once seen as a rival to selection, actually supplied the raw material for natural selection by creating genetic diversity. 41-42. A second synthesis. Evolutionary developmental biology relates the evolution of form to the precise pattern of gene activity, here gap genes in the fruit fly, during embryonic development. 43. Ernst Mayr recognized the key importance of reproductive isolation for speciation in his Systematics and the Origin of Species, 1942. 44. W. D. Hamilton conceived of kin selection in 1964. 45. This synthesis cemented natural selection as the foundation of evolutionary theory, where it remains today. A second synthesis was brought about at the end of the 20th century by advances in molecular genetics, creating the field of evolutionary developmental biology, Evo-Devo, which seeks to explain the evolution of form in terms of the genetic regulatory programs which control the development of the embryo at molecular level. Natural selection is here understood to act on embryonic development to change the morphology of the adult body. 47, 48, 49.